one. Alright, I'm Wai Ling, Dr. Wai Ling from Online University, and the question is directed to Dr.
Mai.
Dr. Mai. Okay. So it's very interesting that you developed the resistant line in the lab, right? And then I just wonder because it could be many directions of evolution in the lab as well. I know the common phenotype is you won't die after killing the velvet ground. So I just wonder if you already done some checking of the phenotype of the resistant line, whether or not it's actually tally with what you have found from the PGCA database, because that's the first study
Yes.
I'm just curious, like,
Yes.
whether or not the in vitro
Yes,
analysis
this project eventually, we thought of that as well. So actually, there is a second project that is already got FRGS. So they are sequence all these CTL cells. So we have the sequence already, and there's another PhD student is working on that. I think looking at, she's actually sitting there. Yeah. So that's been into another project. Actually, we already sequenced all this. We already. And we are now looking at the differential expressions to reconfirm back the understanding. Yes, the short answer is yes. And also, just to let you know, because we started with pancreatic cancer, we also did the same thing for MPC. So we have the MPC cytotoxicity cell resistant line. So that's going to
yes,
the...
here means is the resistant line, the phenotype is mapped
with the,
with
the expected data, yes. It is, but we are going into deeper to understand beside on the HTAG what else is there.
So you only have one clone have been developed in vitro.
We use several cell line.
Right.
Yes, each cell lines we have one clone, the best clone. Yeah. Yes.
Yes. Yes.
Hi, I'm Chung from QI of Australia. This is maybe targeted to Prof. Goh first, and then I also have a follow-up one for Prof. Yap, if it's possible. So for Prof. Goh, how do we access your toys?
Have you emailed us?
You can contact me straight as well and email us.
So is it like through, if we were have to...
Two mode of corporations.
Yeah.
So be frank, I think we do understand that from academic or scientific findings and everything, it's always back to the context of who owns the data, right? So if let's say you seek for the ownership fully, then we will seek, you need to seek for is like from our part is a full service. So if let's say you seek for collaboration, then the fees is minimal. Yeah. So it's very straightforward in that sense. So in that way, actually, just for information, like we have MMR, X-ray crystallographies, everything. We receive the samples far reach even from Pakistan. Yeah. So sometimes, so it's up to you what you want to use, and we can explore together. Yeah.
So I guess my real question for Prof. Yap is that I personally, as a geneticist, I'm always very interested with underrepresented population. Obviously, NPC is one of the disease that's very common in the Bidayo and the underrepresented population in Sarawak. From a traditional geneticist point of view, we are actually very interested in understanding things like founder effect, and so I guess my question is whether there had been any documentation from these underrepresented ethnicities of any kind of ancestry or pedigree that you can map, like like the Chinese way where you can map 3 or 4 generations. And do you know if there's anything in this space, and is there anything, for example, linked to like mitochondrial DNA to track that? Because most of the disease that we see where there's a very strong bias in certain ethnicity, they somehow date back to like a common founder effect. So it's just keen to know a bit more about that.
Yeah. So this is the question when we attend conference overseas. The international researcher always asks us why, and we Malaysia we have a group. I think Susan. Okay. So she got a shock. But we have a group in Malaysia is doing a GWAS study, on this, and then we will wait for their publication. So what I would say that, of course genetic will play a role, but I'm not sure whether Bidayu and Chinese, we can say they are definitely different, because there are a lot of mismarriage, and we all like Asian. But what we notice, and through the communication with the people in Sabah and Sarawak, I think the environmental and dietary actually play a major role in Sabah and Sarawak. And we did an outreach activity in Sarawak recently, and I think the people, the concept, they don't want to know about cancer. They don't want to associate themselves with cancer. So the late presentation actually is quite bad over that, because they don't want, yeah, they don't want to say that, okay, I got cancer. Yeah. So most the late presentation in Sarawak is worse, much worse than the Klang Valley region.
I see.
Yeah.
So in that case, does that mean the manifestation of this disease is very different from southern China compared to the Bidayos or the Iban? Because if that's the case, that means the environmental stimuli will have to be exactly the same for them to have high prevalence, right?
I wouldn't say that is a very significantly difference, because the genetic, I think that we can't change. I think that, okay, my personal view is it will be more or less quite similar. And then their tumor characteristic is similar. Majority are very similar. And the dietary and environmental, actually you can, because EBV infection is more than 90% of the population, and then why somebody got NPC, some don't. Yeah. So what we know that is the insult from the environmental. Let's say maybe the thing that you eat, or the air pollution actually will trigger some pre-malignant changes in a certain genes, like P16 or cyclin D1. Yeah. So it already been shown that cyclin D1 can it support the latent infection of EBV in the cells. And our group and also show that TGF-beta R2 loss is also help to support the latent infection of the virus in the epithelial cells. So it's a multifactorial thing. And then I would just say that in China, Hong Kong, and Singapore, the incident rate of NPC actually has gone down. Yeah, because they eat more healthy.
I see.
Yeah. So this is what we would like to promote in our country. Eat healthy. Yeah.
Thanks.
Okay. I have a quick request. You want to add on something, Prof. Goh?
Yeah. So just add on just now a question. So why just now actually I showed 13 groups of businesses for Sunway. So actually I'm trying to show you the positions, how we position ourselves. So within the university, we receive the samples from engineering schools as well, a lot. So we do a lot of material science analysis, even the carbon dioxide capture analysis, everything in our lab. And this within university. Within group, we serve for Sunway Multicare. I think Prof. Felicia also helping for clinical trials as well. So, and within state government in Selangor state, we actually is part of lab partnership. So this actually, those companies register under SME in Selangor, they can send samples for us to do analysis. They claim back from state government up to 5,000 per year. So just, I forgot to add on this now. So just want to add on that. Anyone actually really need the access, no problem. Yeah, thanks.
Yeah, for the SMEs, looking at them. Thank you. I have a quick question for Prof. Yap actually. So following on what you mentioned to Liu Sheng's question, the tumor characteristics are very similar. You were saying it did, and you also showed it that the transcriptome is also very similar. Is it between the Bidayu and the non-Bidayu, or the Chinese versus the non-Chinese, basically? Which is very interesting because they have such a much higher incidence, and yet the transcriptome is the same. And then you were mentioning that the pathways, the cyclin D1 and TNF, was it, or TGF? Yeah, is a highly relevant, highly associated with the incidence. But in the Bidayu, yet you don't see this being extremely high, do you? Or do you see it being extremely high compared to the Chinese cancers? Because if the transcriptome is the same, then how is I'm trying to wrap my head around why it is that these two pathways are, if you consider it to be associated with the incidence, why is it not increased or exacerbated in the Bidayu or Iban?
Okay, so this P16, cyclin D1 and TGF-beta is not associated with the incidence. It's the pre-malignant changes
I see.
that support the latent
Right.
infection of EBV in the epithelial cells.
I see.
Yeah, so it's different.
Right.
And because of the time constraint, so I did not show all my RNA-seq data. So we saw some changes that are predominantly in Bidayu. We did some, but we need to do more validation about that. Yeah, so it's still, currently we still focus on those that commonly are deregulated.
Right. Even the EBV transcript, the EBV genes are not the same, not different between the
No obvious difference.
Oh, okay, very interesting.
And then you can see their EBV profiles, EBV antibody profiles are more or less similar.
Okay, kind of good news in the way that you treat
Yeah,
one, you can
yeah, it's a good news
Mhm.
because what I was told was because once I talked to the company which conduct clinical trial in Malaysia, and then I say why you don't go to Sarawak and Sabah? There's so many patients. They say because we don't know whether they are the same as Chinese, so they don't want to go there. So I thought this is not right.
Yeah, very important.
Yeah, so I did this study just to prove that they are the same. So if you want to do study, please include them. Yeah. So
Okay. Taking a quick look around. Are there questions, or I will misuse my chair privileges to ask questions more more questions? Then I have a question for Prof. Goh. Actually, a few. So you were mentioning talking about your EV work. I was curious, like when you look at the EV work, what inside the EVs are you looking for? You mentioned the highly volatile compounds. Is there like DNA, RNA, anything else inside siRNA?
Want to try?
Outsourcing.
Actually, we're working with the University of Bogota, right? So they actually developed the biosensors on the eNose. So eNose, if let's say we talk about it, it's like graveyard of science because the way of detection is so tough. Actually, we know that we can use the GCMS or GCFID to look at some of the volatile components that is conventional, but in terms of response with the specific compounds, that material they synthesize is different. So 12 sensors being built to look at the volatile components evaporate from the urine. But we streamline, work together, we get the EVs because from FDIR we're able to see the changes for prostate cancer, right? So we are just thinking that using the eNose, could we detect any volatile components out of there rather than conventional genomic proteomics or whatever proteins or whatever things. So it's something totally different. Yeah, so volatile.
Yeah, it's interesting that the EV can contain the
yes,
volatile
very
compounds.
little different. So we synthesize the chips at Romania, right? So MGM over there. So we are just trying. So MGM is just a company in Romania that actually preparing components for CERN in Switzerland, electron collider. So they produce the components for them. And we went there, they actually did the some of the chips for us that you can do the EVs. Then after that, the volatile components will detect by chips and amplify the signal. But I think the project is rather not easy, long process, a lot of challenges. Yeah, so we facing a little bit of problem on the, I think the threshold detection as well. It's workable with, if let's say the urine, I think 5 mil or 10 mils there. Yeah. So then the volatile components you can straight away detect. But now we purified the EVs, the detection threshold gone down very much, a lot. Then the chip sensitivity, now we try to amplify the signal. Those kind of things are still ongoing.
I see. Then if you don't mind me asking, to train the AI model, because in the same slide you have the AI model, how many samples are you looking at?
It means
For the pancreatic cancer,
for the FDIR, I think we are collecting 300 at the moment for minimal.
300 cancer
Cancer patient.
and
prostate.
Prostate cancer
Yeah.
patient.
cancer, right? And then versus normal 300, is it something like
No,
this?
total is
Total.
300.
Oh.
So far.
Minimum. Yeah.
Oh,
Yeah. Minimum. Or else you can't train. Yeah. Minimum is
Yeah.
300.
okay. Right, okay. Can,
On and on.
alright. Yeah, then my next question is for the both of you, Prof. Goh and Prof. Mai, since you are both working on that project, and you mentioned Prof. Mai in that project, the one where you have your pipette go over to the receptor and deposit the ligand. How do you measure that that receptor is activated?
Okay, we actually have a discussion because SICM basically it's not the like if let's say confocal or whatever thing is like chip based or scanning.
Yeah.
This one is like it will based on the structure and move along the way. So we are actually in the discussion because it bound with the fluorescent microscope. So we are just thinking that can we use the fluorescent signals as well to detect that receptors first, so that it will be guided over there, then only release the minute amount specifically on that receptor, that area. Yeah. That is what we planning to discuss. I think on 26 you are in that. But I told the staff already. Yeah. So that is what we how to put it. Because the technology is the things that we are going forward for, but we need to try out as well. So that's why the companies allow us to send the samples and doing the testing over there. So how to start with anything in our hand, then we bring it up and do it. Yeah, and so happened that we work together on it. Then that's how the whole thing's developed.
Actually, I was curious, but how do you know the receptors being activated? It can change. It will detect the change in structure, or
You use the antibody fluorescent signal, then you will know the receptors.
it will detect the receptor. Then it will go there and deposit.
Then you see the cell response.
Oh, then you have to wait for the cell response.
Yes.
Okay.
Yes.
But sometimes they might have electrode changes. So those are.
Okay, you want to look at the changes because the electrode itself, you can load the ligand, but on the same time, electrodes, that pipette actually can study the microenvironment, pH and everything changes at that region. So it really depends on the parameters that you want to look at it. So in short, at the moment, if let's say changes is there, any parameters we can detect, it make meaningful for us at the moment because we haven't tried it yet. So in short, we are seeking forward for either it's positive or negative data. Positive, then we can celebrate.
Unless you said that antibody, unless the antibody binds nicely to where the ligand will be, and the ligand will kick it off. I'm not sure if that's
Be frank, we're trying out at the moment, but the idea is roughly in that manner. 26 meeting.
Seems like I just
Pray
Are there any questions from the floor? If there are no additional questions, then thank you very much to the panel. I will be